All right, we can go ahead and get started, and some other people will trickle in, I'm sure. Um, Welcome back, everybody, to the colloquium series for the fall semester in the Department of Anthropology at the University of New Mexico. Today, we're really lucky to have a really excellent and special final speaker to end the semester in a really strong way, Dr. Amanda Leah. Amanda was in undergrad at UCLA, where she received a degree in Ecology and Evolutionary Biology. She then went on to complete her PhD at Duke in ecology, where her supervisors or advisors were Susan Alberts and Jenny Tung. And she is currently a Helen Hayes Whitney Foundation postdoctoral fellow at Princeton University. In 2021, she will become an assistant professor in the Department of Biological Sciences at Vanderbilt University. Broadly speaking, Amanda's research focuses on the evolutionary and genetic basis of how human and primate phenotypes are influenced and shaped by social and ecological stressors throughout life. And she's particularly interested in how such stressors affect phenotypes related to health. Her research has involved a lot of really cool field work. During her PhD work, she worked with yellow baboons in 𝗵𝗶𝗀𝗶𝗃𝗃, Kenya. As a postdoctoral fellow, she has worked with the Turkana people, a pastoralist population living in northwest Turkana near Lake Turkana. And Amanda has also been one of the key members in developing a new research project with me and some other people in Malaysia called the 𝗭𝗿𝗍𝗄𝐿 Health and Lifeways Project, working with the 𝗭𝗿𝗍𝗄𝐿 people of Malaysia. And even though Amanda is pretty early on in her career, she has published very widely in a lot of excellent journals, exclusively in excellent journals, including journals like PNAS, Proceedings B, eLife, Science Advances, and many others. I can't express how highly I think of Amanda and her work, and how thankful I am that she is willing to speak with us today and end this fall colloquium seminar series in a really strong way. So thank you very much, Amanda.
Great. All right, let me get my screen sharing. Okay. Okay, can you guys see my full screen?
Yep.
Okay, great. Well, yeah, thank you very much, Ian, for that kind introduction, and thank you all for being here. I'm really bummed I wasn't able to come in person, but I'm glad at least that I can give this talk to you and that we can chat a little bit afterwards. And so today, as Ian sort of alluded to, I'm going to tell you about work that I've been doing as a postdoc looking at genomics and at health in the Turkana people. And so this work is motivated by the fact that cardiometabolic diseases are a major global problem. To illustrate that point here, some plots showing worldwide trends in BMI over the last few decades. And we can see that BMI has remained high in high-income countries such as the US. And in addition, BMI is increasing rapidly across the world as countries become more urban and more industrialized. And so why is it that industrialization and urbanization seem to go hand in hand with cardiometabolic disease? Well, one theory is that human physiology is poorly equipped to deal with urban industrialized lifestyles. And in particular, there's a hypothesis from the field of evolutionary medicine known as the evolutionary mismatch hypothesis, which posits that traits that evolved in response to selection pressures in the ancestral environment of a population may no longer be advantageous if the environment shifts and individuals find themselves in a novel environment. And with respect to cardiometabolic disease specifically, the idea here is that essentially all human populations evolved eating a mixed plant and meat-based diet that took considerable energy to obtain. And through evolutionary time, we think that our genomes and our physiology adapted to this type of diet and lifestyle. However, in modern societies today, we see a very different situation where a much higher in fat and carbohydrate diet can be easily obtained through a sedentary lifestyle. And therefore, it's been proposed that our genomes and our evolved metabolism are mismatched to our current environment, which is what's leading to cardiometabolic disease. And the evidence that's commonly used to support this idea comes from health comparisons between high-income countries and small-scale subsistence-level groups who are arguably living lifestyles that are more consistent with their recent evolutionary history. For example, here's a summary of data from Eaton et al. Showing that type 2 diabetes prevalence across a number of modern-day subsistence-level groups, including hunter-gatherers, horticulturalists, agriculturalists, and pastoralists, is about 10-fold lower than what we see in the US. And these results from Eaton et al. Have now been followed up on by numerous groups, and there are many papers of this flavor out there to show that small-scale subsistence-level groups tend to be in better cardiometabolic health relative to fully industrialized high-income countries. And this body of work has been extremely influential, and it seems to be consistent with the idea of evolutionary mismatch. However, I would argue that it has a few limitations and a few gaps that we'd ideally like to fill. The first gap that I see is that the predictions of the evolutionary mismatch hypothesis are that individuals from a given locally adapted population will do better in the environment in which they evolved relative to a novel environment. However, when we compare subsistence-level groups versus the US, we're actually contrasting two populations that have pretty different genetic backgrounds and very different recent evolutionary histories. And consequently, I don't really think that we're addressing how a single population would perform in their ancestral environment versus a novel environment. We're instead just comparing two separate populations. Another problem that I see is that the sorts of health comparisons that I just described are rarely, if ever, complemented with genomic work in the same subsistence level groups. And so it's unclear to what degree these groups actually have genetically adapted to their traditional life ways, even though this is a major assumption of the evolutionary mismatch hypothesis. And then finally, again, because there are so few data sets that combine genomic data with health and lifestyle information in subsistence level groups, there have been very few attempts to actually map what are known as genotype by environment interactions as we would expect them under evolutionary mismatch. So in particular, we don't have many examples of loci or variants in the genome that look like this, where if you carry in this cartoon the A allele, you're always pretty healthy. But instead, if you carry the T allele, your health is really dependent on the type of environment that you find yourself in. And more specifically, in this example, the T allele has positive effects on health and fitness in the ancestral environment, but this same allele then becomes associated with disease in a novel or modern environment. And while this sort of cartoon example of a genotype by environment interaction is a pattern that would be consistent with evolutionary mismatch, it's been argued by Greg Gibson and by others that this particular pattern might actually be relatively rare, because we expect that over evolutionary time, stabilizing selection and purifying selection should limit the number of loci in the genome where genetic variation is associated with health or fitness outcomes in the ancestral environment of the population. And therefore, a more common pattern might be that there's genetic variation that's simply neutral and not associated with health or fitness in the ancestral environment of a population, but then that genetic variation becomes associated with disease when the population is exposed to a novel or a modern environment. And so over the last few years, I've been working to develop a study system that would allow us to overcome these gaps in our understanding of evolutionary mismatch and to more directly test this idea. And that work has been focused on the Turkana people. And so first, I just want to give you a little bit of background about the Turkana and sort of convince you that I think they're a good model for addressing the questions that I've laid out. So the Turkana are traditionally a small-scale subsistence level group from the arid regions around northwest Kenya, where they've lived for at least the last several thousand years. During this time, they have remained relatively isolated, partly because this region of Kenya is extremely inhospitable. The year-round temperatures in this region are in the 90s, and there's very little rainfall, as you can see on this plot of average climate throughout the year. Given that the environment is so arid and so inhospitable, there are pretty limited options for water. And to cope with this water scarcity, the Turkana drink relatively little water each day, which they source from deep boreholes that are dug in dry riverbeds and which they walk long distances to obtain. The aridity of this region also means that there are pretty limited options for gathering or for cultivating food. And consequently, the Turkana people practice nomadic pastoralism, and they rely on livestock that they herd and keep with them for subsistence. As a result of this pastoralist lifestyle, the Turkana consume a diet that's very meat and protein centered. And about 80% of their diet comes from milk or milk products, which is shown on this caloric breakdown in dark blue, or comes from red meat, blood, and other animal products, which is shown on this breakdown in light blue. And so this is what the diet and the ecology and the lifestyle of the Turkana has historically looked like. And these are the sorts of environmental challenges that we think they're locally adapted to. However, over the last few decades, things have really started to change. Infrastructure has increased in this previously remote and isolated region, partly as a result of oil discoveries in South Sudan. And consequently, many Turkana have now stopped practicing subsistence-level nomadic pastoralism, and they now live a much more sedentary and market-integrated lifestyle that's focused on trading items such as woven baskets, fish, and charcoal. And in addition to the emergence of this group of people that I'm going to call non-pastoralists, who still live within the relatively remote and rural Turkana region, there's also a group of Turkana that have begun to leave both their traditional homelands and their traditional pastoralist practices entirely, and are now moving into densely populated areas in central Kenya. And in these densely populated areas in central Kenya, they experience a much more Western-like diet and lifestyle. And so over the last 3 years, my postdoc advisor, Julian Arrols, and I, in collaboration with 2 Kenyan scientists, Dino Martins and 𝗭tože𝗵𝗃, as well as a really amazing field team pictured here, have been collecting data from across this lifestyle gradient spanning these 3 different groups. Namely, we've been sampling traditional pastoralists in the rural and remote Turkana region. We've been sampling people that have stopped practicing pastoralism but still live within that remote Turkana region, and then people who are living a fully urban lifestyle in central Kenya. And as of April, before COVID shut down our research, we had sampled about 2,200 people across the 57 locations shown on this map. In each of our sampling locations, we conduct in-depth interviews with our study participants. So we ask them about their diet, about their occupation and their subsistence activities. We collect a health history and a reproductive history, and we also ask about early life experiences and about where people have lived throughout their life, because we're interested in sort of a life course perspective on health. We also collect data on 9 common measures of cardiometabolic health. Namely, we measure waist circumference, BMI, body fat percentage, we run a lipid panel, we measure blood glucose, and we measure blood pressure. And finally, we also collect biological samples. Namely, we collect a couple of different types of blood samples. We also collect serum, urine, and fecal samples. And before I tell you about the scientific questions that we've been using these data and samples to ask, I just want to say a few words about how we work with these Turkana communities, since I view this as an important part of the methods. The communities that we work with are relatively hierarchical. So when we're interested in working in a given location, we first approach the chief and we discuss our project with him. If he thinks that his village might be interested, then we have a larger meeting with the local elders. And if that goes well, then we have a meeting with all community members. Before we do any sampling at the individual level, the study is also re-described to the participant in their preferred language, and it's emphasized to them throughout all of these meetings and during their individual consent that they can opt out of any parts of the study at any time. It's also important to us that the communities that we work with and that Kenya more broadly benefits from the work that we do, and we therefore make community level contributions in the villages that we work in. We also always have local nurses on site who can consult with our participants on any health-related questions that they might have. And then during the last few months and sort of during the time of COVID, we've been working to develop and distribute COVID educational materials to the Turkana communities that we work with that are specifically targeted to the pastoralist context. And also over the last few years, we've been working with Impala Research Center in central Kenya to build more capacity for in-country genomics research. And we've been doing that by building a molecular lab at Impala and by offering training in molecular techniques. And I'm happy to talk about any of these aspects at the end of the talk. Okay, so now we've got this data set of paired interviews, biological samples, and health biomarker data. So what can we do with it, and how can it help us understand evolutionary mismatch? Well, because we have this dramatic lifestyle gradient within a single population, we can ask how health differs as individuals' lifestyle changes from matched to mismatched. And because we've collected biological samples, we can also use a whole genome sequencing approach to test for local adaptation to the population's traditional lifestyle. And then finally, we can use our paired genomic and phenotypic data to try to actually map genotype by environment interactions. And I'm going to start with this first problem of looking for lifestyle effects on health. But before I show you any of the health data, I just want to show you a little bit of the metadata and the interview data to convince you that the three lifestyle groups that I described really do follow along a gradient from traditional and sort of matched to the evolutionary history of the population to more urban and mismatched. And that appears to be true if we look at one commonly used measure of urbanicity on market access, which is distance to a major city across the three lifestyle groups. You can see that that follows a pretty linear trend. In addition, if we look at dietary items that are commonly consumed by the three lifestyle groups, we can see that almost all traditional pastoralists shown in light blue report that they regularly consume blood, milk, and meat as expected, and they consume very few carbohydrates or food additives that would be obtained from the market economy. In contrast, individuals that are living in urban areas, which are shown here in light green, report that they regularly consume a number of different carbohydrate products, and they use additives like salt and sugar in their cooking. And individuals that are living in that remote and rural Turkana region but not practicing pastoralism fall somewhere in between these two extremes. And so now we want to know whether those lifestyle differences translate into differences in our health biomarkers. And to ask that question, we simply looked across each of our 9 biomarkers and asked whether there was a difference between each pair of lifestyle groups. And I'm about to show you a plot where on the x-axis is going to be the magnitude of the effect for the comparison between a given pair of lifestyle groups, with larger positive values indicating higher biomarker values in the more mismatched group. And translucent bars are going to represent contrasts that were not significant. And so here's what those results look like. And what we can see is that there are consistent health differences between pastoralists and urban individuals, which are the contrast shown in blue, and between rural individuals that are no longer practicing pastoralism versus urban individuals, which are the contrast shown in green. However, we don't actually observe any health differences between pastoralists and rural individuals that have stopped practicing pastoralism, which are the red bars, which are all translucent. And so to summarize what it seems like we're seeing, there are health changes in the Turkana as a function of lifestyle, but it seems like these effects are nonlinear, and you have to get really far along on this gradient of mismatch, which is what we see in that fully urban group, for there to be any changes in our biomarkers. And so after seeing these results, we had a number of follow-up questions. We wanted to know how bad the biomarker changes in the urban group were that we were seeing in terms of disease risk. We also wanted to know why these changes were happening, and we were especially interested in the role of diet. And then finally, we wanted to know when in an individual's life exposure to these urban environments seemed to actually matter. To tackle this first question, what we did was to just take cutoffs from the CDC and the American Heart Association that are commonly used to diagnose disease. And we asked how many individuals in both the urban group and the rural group reached these thresholds, which is what I'm going to plot on the y-axis. And when we do these calculations in the Turkana, and here I'm also adding data from the US for comparison, we can see that urban Turkana are much more likely to reach thresholds that put them at risk of cardiovascular disease relative to the 2 rural groups, which I've grouped together here. But this risk hasn't quite reached the levels that we see in a fully industrialized and high-income country, but it does seem like the differences that we're seeing are potentially meaningful. To address our second follow-up question of why we were seeing biomarker changes in the urban group, with a particular focus on the role that diet might play, we turned to our interview data. And what we did was to just take each of the dietary variables that I showed you a few slides ago, as well as a few other variables that we had data on and that we thought might be important. And then we used a technique called formal mediation analyses, which just allows us to estimate how much of the lifestyle effect on a given biomarker is explained by a given potential mediator. And here's what the results of those analyses look like, where what I'm showing you on the x-axis is each of the potential mediators that we tested, and they're broken down by sort of broad scale categories. And they're colored in by the magnitude of the effect of lifestyle on health that they could explain for a given biomarker. And what I want you to take away from this plot is that shifts away from a traditional pastoralist diet and towards a more Western-like diet that incorporates carbohydrates and processed foods does explain some of the variance, especially for waist circumference and BMI, but clearly changes in diet are not the only important mediating factors, and they certainly don't explain all the variance. Okay, and then lastly, our third follow-up question was that we wanted to know whether an individual's exposure to an urban lifestyle mattered across the lifespan, and in particular, we were interested in early life experiences. And we can ask this question in the Turkana because there's currently a lot of within lifetime movement in this group. To illustrate that, here's a plot showing the birth location for each participant in our study, colored as a red dot. And that's connected then to the location where we sampled that individual by a gray line, with the sampling locations outlined in black. Just to emphasize this variation in another way, here's a plot with the measure that we're going to use to define the early life environment, namely population density of each individual's birth location. And I've broken that down by individuals that were sampled as adults in the remote and rural Turkana region in green versus individuals that were sampled in urban areas in central Kenya in blue. And what we can see is that individuals born in low population density areas tend to fall in the rural lifestyle group as adults, and similarly for urban individuals. However, early life and adult conditions are definitely not collinear, and we can use that variation to tease apart the independent effects of early life and adult exposures to urban environments. And when we do this statistically, we find that there are 3 biomarkers where early life population density, which is shown here on the x-axis, has effects on traits that are measured in adulthood while controlling for adult lifestyle. And in particular, we can see that being born in a more urban and high population density area is associated with higher adult BMI, higher adult waist circumference, and higher adult blood pressure, no matter what your adult lifestyle looks like. And I think that this result is really important because it suggests that urban environment effects on health are going to be additive and they're going to accumulate across the lifestyle, which really argues for taking a life course approach to lifestyle effects on health. All right. So now that we'd seen some clear within population variation in health as a function of lifestyle, we wanted to know whether this could be explained by a locally adapted genome encountering a novel environment. And so to answer this question, we've been collaborating with Sarah Matthew, Ann Stone, and Melissa Wilson's groups at ASU. And in collaboration with this team, we've been using blood samples combined with high throughput DNA sequencing to generate a data set consisting of 301 whole genome sequences from the Turkana, as well as 56 additional whole genome sequences that we've collected from 8 other groups in this region. And the 8 other groups that we sampled are groups that live near the Turkana and are potentially thought to share some sort of evolutionary relationship with them. However, because there's almost no existing genomic data for this part of Africa, those relationships are very poorly understood. And so one of the first things that we wanted to do was to ask what genetic variation looks like in the Turkana relative to other African groups. To do this, we downloaded publicly available sequence data from the 1000 Genomes Project, which has generated whole genome sequence data for a number of different African groups, focusing especially on West Africa, but providing data for one additional Kenyan population, the 𝗭𝗏. And we then merged our data with this 1000 genomes data and used principal components analysis to ask how all these different groups were related to one another. And when we did that, we got results that looked like this, where on this plot, each dot represents an individual genome, and it's colored by the population that that individual came from, with the colors corresponding to the map on the left. And the result that immediately jumps out at us from this PCA analysis is that the first principal component seems to pretty clearly separate all of the West African samples from our Turkana samples, with the 𝗭𝗏, that other Kenyan population, falling somewhere in between, but actually closer to the West African samples. And it turns out that this separation along PC1 actually makes some sense because the West African groups and the 𝗭𝗏 all speak languages that are classified as Niger-Congo, whereas the Turkana and the other groups that we sequenced speak Afro-Asiatic and Nilo-Saharan languages. And therefore, PC1 seems to be separating out one of the major language families and sort of the known lineages that exist within Africa. We next turned to PC2 and looked specifically at the loadings for R samples, where that variation seems to be concentrated along PC2. And so now I'm showing you on the left the principal component 2 loading for each individual in our data set, which is on the y-axis, and that's stratified by the particular population that each individual came from. And we can see again that there seem to sort of be 2 clusters here, with the Turkana, the 𝗵𝗏, the 𝗔릿𝘁𝗵𝗿𝘆, the 𝗵𝗏, and the 𝗖𝗏 loading high on PC2, and the remaining groups loading lower. And in this case, rather than reflecting language families, the separation seems to actually reflect geography, such that the Turkana seem to be most genetically similar to other groups that live in close proximity to them on the west side of Lake Turkana and in sort of this northwest corner of Kenya. And we're currently in the process of doing more formal demographic inference on this data set, but hopefully this at least gives you a glimpse into what genetic variation in this region of East Africa looks like. All right, so now that we had some sense of our data set, let's turn to this question of local adaptation. To answer this question, we're going to specifically look at the 301 whole genome sequences that we generated specifically from the Turkana. And we're going to use this whole genome sequencing data set that consists of about 11 million variants to identify regions of the genome that are putatively under positive selection. And we went about this in 2 ways. One approach was to look for classic selective sweeps at individual loci. And this happens as a pattern known as hard sweeps. And this happens where there's some new mutation that arises in a population, and it then sweeps to high frequency very quickly. When this process occurs, it leaves behind relatively distinct patterns of haplotype structure and of allele frequency differentiation between the focal population and any other populations. And we can look for those sorts of obvious patterns in genomic data. And while these sorts of single locus hard sweeps are really interesting, and they've taught us a lot about human evolution, a process called polygenic adaptation, where there are many small and subtle shifts across many different loci that contribute to a trait, is actually thought to be a much more common type of evolution. However, this pattern is much more difficult to detect in genomic data, but if we have some idea of what phenotypes we think are putatively under selection, there actually are some tools and some approaches that we can use. And so in our case, we hypothesized that cardiometabolic and renal system traits would be under selection due to the Turkana's unique high protein diet and due to the fact that they're living in this arid desert region and they're extremely water stressed. And so what we did was to download genome-wide association summary statistics for 15 different cardiometabolic and renal system biomarkers that have been quantified in consistent ways and across diverse ancestries in this very large study called the UK Biobank. We then calculated 3 different statistics that quantify positive selection for every locus in the Turkana genome. And then we simply asked whether regions that are known to be associated with a given biomarker from that genome-wide association data that we got from UK Biobank, we just simply asked whether those regions showed more evidence of positive selection in the Turkana relative to randomly drawn regions of the genome. And when we did this, we find that many renal system and cardiometabolic biomarkers seem to potentially be experiencing polygenic selection in the Turkana, such that the aggregate evidence for selection that we observe across all these different regions, which is the values plotted in red, is greater than null expectations, which are the distributions plotted in black. And in particular, I want to call your attention to the renal system biomarker at the top, urate. Urate, or uric acid, is a waste product that is produced and eliminated by the kidneys when the body breaks down purines. And these purine molecules are actually found in really high concentrations in red meat and in organ meat, which we know is a major component of the Turkana diet. Okay, so now we've got some interesting evidence for polygenic adaptation on phenotypes that are potentially meaningful for the Turkana. So now let's ask about this other pattern where we're looking for strong selection at a single locus. To look for these cases, we simply asked whether there were regions of the genome with more genome-wide outlier loci than we would expect by chance across all three of the different selection statistics that we calculated. And when we do this, we come up with 5 regions that are putatively experiencing selective sweeps. In the interest of time, I'm just going to focus on the most significant region that we think is our best candidate for positive selection. And I'm zooming in on that region now and showing you about 200,000 base pairs on chromosome 8. And so in each of these plots that I have here on the left, what's on the x-axis is the location in the genome, and each dot represents a variant that we tested. And on the y-axis is the effect size for each of the 3 different selection statistics that we calculated, with the 95th and the 99th percent or genome-wide percentiles noted in light blue and dark blue. And so what I want you to take away here is that there are many outlier large effect loci in this region across all 3 statistics. There are many loci with large effects relative to the genome-wide distribution. And so what is this region? Well, it's a putative regulatory region that's upstream of a gene called 𝗭𝗵𝗃1. And this 𝗭𝗵𝗃1 gene has many different functions in the body, but it's particularly important for maintaining ion balance in the kidneys. In genome-wide association studies in humans, 𝗭𝗵𝗃1 has been repeatedly associated with glomerular filtration rate, which is just a commonly used measure to assess how well the kidneys are functioning. It's also been repeatedly associated with uric acid levels, which I just told you are produced when the body breaks down purine-rich foods such as red meat. In rodent experiments, 𝗭𝗵𝗃1 has also been shown to be involved in glucose homeostasis, such that when researchers infuse human 𝗭𝗵𝗃1 into the medulla region of rodent kidneys, this actually alters the glucose production in these rodents. And then finally, in addition to these sorts of metabolic functions that 𝗭𝗵𝗃1 seems to have, it's also known to be involved in the response to dehydration. And in particular, again, in rodent experiments, if mice are deprived of water, 𝗭𝗵𝗃1 is strongly induced, and the relationship between 𝗭𝗵𝗃1 induction at the level of gene expression seems to change linearly with the amount of water deprivation that these rodents are exposed to. And so altogether, this is extremely exciting, given that I told you that the Turkana live in a really arid region and that they're extremely water stressed. I also told you that they subsist on a diet that's very high in red meat. And so it appears that we are finding signatures of these ecological challenges in their genomes most strongly in this regulatory region near this 𝗭𝗵𝗃1 gene. And so right now, this is really shaping up to be a pretty compelling story, but there's still a lot of work for us to do to validate that the genetic variation that we see in this candidate region actually has physiological effects in the Turkana themselves. And so to do that right now, what we're doing is to take the serum samples that we've collected from the Turkana and to use mass spectrometry to measure a handful of metabolic and renal system biomarkers in the Turkana samples. Using these data, we're then performing focused association mapping in our candidate region to ask whether we can identify variants in this region that are associated with any of these biomarkers. And at the moment, we've generated preliminary data for about 138 individuals, which I'm showing you here, where on the x-axis is just a location in the genome, again, in that candidate region that we're interested in. And on the y-axis is the magnitude of the effect for an association between genetic variation and glucose or uric acid levels. And so excitingly, we are seeing some associations in this region that would pass our genome-wide significance criteria. And currently, we're in the process of adding more individuals to this data set and adding a few more molecules. And so we're hoping that these results hold up. And if they do, then the next step that we're going to take is to take the variants that seem to have significant genotype-phenotype associations in the Turkana and take those into an experimental molecular assay known as a reporter assay, which will allow us to actually causally test whether these variants alter regulatory activity in our tissue of interest, which in our case is the kidney. Okay, And so now that we'd established that the Turkana are putatively locally adapted to their subsistence level lifestyle, and that when individuals of this genetic background are placed in a very different set of conditions, namely in an urban environment, their health suffers. We wanted to address this third missing piece of the puzzle about mapping these genotype by environment interactions. And specifically, what we want to know is whether there are genetic variants that are advantageous or neutral in the ancestral environment of this population and in the pastoralist environment, but then become associated with poor health in an urban environment. And I just want to stop and acknowledge for a minute that while we think these sorts of genotype by environment interactions are probably widespread and really important in human health and in human populations in general, finding them is incredibly hard, and very little progress has actually been made on this point in any population. And that's because naively, to find loci that fit the pattern that I have here on the left, what I need to do is to do a genome-wide association study for a trait like cholesterol or for a disease like type 2 diabetes in Turkana pastoralists and in urban Turkana, and then identify loci that are associated with the trait in one environment but not in the other. And doing this sort of genome-wide association mapping is notoriously hard and difficult, and it takes huge sample sizes that we just aren't going to get right now. And so we need to get creative about this problem. And to do that, I've been taking two different approaches. The first approach that I've been taking is to do the sort of mapping that I just described, but instead of putting cholesterol levels or type 2 diabetes on the y-axis, I'm using a more tractable and intermediate molecular phenotype. In particular, I've been working to generate genome-wide genotype, gene expression, and DNA methylation data for individuals that are living a pastoralist lifestyle, as well as individuals that are living an urban lifestyle. And I'm able to then use these data to map genotype by environment interactions that would fit the pattern that I've shown here. But I can do this with more power than I would have if I was using a downstream biomarker like cholesterol levels. And I'm actively in the middle of doing this work right now, and so I don't have the results to share with you quite yet. But in parallel with doing this sort of molecular phenotype mapping, I've also been taking a second approach, which is to use a model organism where I can get the large sample sizes that I need to do the sort of more organism level trait mapping. And in particular, I'm going to tell you very briefly now about some work that I've been doing in fruit flies to map genetic effects on lifespan when flies are exposed to a normal standard lab diet versus an obesogenic diet that is very, very high in sugar and that's outside of the range of anything a fly would ever experience in the wild. And while this sort of experimental framework obviously isn't the same as exposing a human group adapted to subsistence level practices to a Western lifestyle, we did pick this obesogenic and high sugar diet in flies as our novel environment of interest because of the central role that diet is thought to play in the evolutionary mismatch hypothesis for human disease, and especially for cardiometabolic disease. To map genetic effects on lifespan in these flies when they were exposed to two different diets, we took an approach that was inspired by some recent work from 𝗭𝗿𝗶𝗄𝗏 and colleagues, in which they mapped variants that affect lifespan in humans. And to do this, what they did was to take advantage of existing data from a couple of very large cohort studies where individuals of different ages had all been genotyped. And for most alleles in the genome, we don't actually expect there to be any difference in how common or how frequent a given allele is as a function of age. However, we would expect this pattern to arise if the allele affects lifespan. More specifically, if there's an allele that actually makes you die earlier, then we would expect that allele to be at higher frequency in younger individuals relative to older individuals, because individuals that carry that allele are removed from the population at earlier ages through death. And so using this sort of framework, 𝗭𝗿𝗏 and colleagues found examples that look like this, where individuals that carry the T allele at a variant near the ApoE gene are more likely to die young, and therefore the allele is at lower frequency in older individuals in the population relative to younger individuals. And so we wanted to harness this sort of intuitive framework, but to also pair it with controlled environmental manipulation so that we could actually map genotype by environment interactions for lifespan. And so in collaboration with another postdoc in the lab, 𝗭𝗨, as well as Peter Andolfato's lab at Columbia, we set up an experiment that looks like this. We took a bunch of wild-caught flies from around Princeton. We then brought them into the lab for about 30 generations to expand the population. And then when we had a lot of flies, a really large population of flies, we randomly allocated age-matched flies at the beginning of their life cycle into 6 different cages with about 10,000 flies going into each cage. In 3 of these cages, flies were fed a standard species-typical lab diet. And in the other 3 cages, flies were fed this extremely high sugar diet that's known to be obesogenic in flies. We then let the flies live out their lives and age from young to old until there were only about 500 flies left in each cage. At that time, we took a sample of all of these 500 oldest flies, and I'm going to call that sample TN. And in addition to sampling the 500 oldest and longest surviving flies in each cage, we also had a sample that we had collected at the beginning of the experiment when all of the flies were young, which I'm going to call T0. And at that time point, we had collected about 2,000 flies. We then took all of these flies from both the T0 and the TN time points and used a low coverage sequencing approach to infer allele frequencies at the beginning of the fly life cycle and at the end. Using these data, we could then ask whether there were alleles that consistently changed in frequency as flies aged, as we would expect if there were alleles that decreased longevity. And in this cartoon example that I have here, I'm plotting an allele that decreases longevity on both diets. However, we can also use this framework to identify alleles that only decrease lifespan on one diet. For example, in this cartoon, the focal allele only affects lifespan on the high sugar diet. And just to give you a sense of what this looks like in real data, here's an example of an allele that starts off at about 70% frequency at T0, so those are the gray bars, but then it's only found at about 60% frequency in flies that live to old ages, with those changes in allele frequency being very consistent across all the replicate cages and across both diets. And now here's a second example from real data, where now I'm showing you an allele with genotype by environment effects on lifespan. This allele starts off at about 40% frequency and then doesn't change significantly in frequency in flies exposed to the standard control diet, but it does decrease in frequency for flies that are on the high sugar diet. And so we tested for these allele frequency changes across the lifespan on both diets. And when we did that, we found that they were actually relatively prevalent. So what you're looking at here now are plots where on the x-axis is all of the different genetic variants in the fly genome that we were able to test. And on the y-axis is the magnitude of the effect for a genetic effect on lifespan on each of the 2 diets. We can then essentially overlap these plots, overlap the results from the 2 different diets. When we do that, we find that there are 1,599 variants that significantly affect lifespan on both diets. There are only 2 variants that only affect lifespan on the control diet, and there are 702 variants that affect lifespan only on the high sugar diet. And so now we want to know whether these loci can tell us anything about evolutionary mismatch. And as a reminder, I told you that there are 2 patterns that we might expect under this hypothesis. There could be alleles that are beneficial on the species typical control standard lab diet, but then become detrimental on the evolutionarily novel high sugar diet. Or alternatively, there might be alleles that are simply neutral on the control diet and then become detrimental on the high sugar diet. If the first pattern were prevalent in our data, we'd expect that for the 1,599 sites that affect lifespan in both environments, on both diets, that their effect sizes would go in opposite directions. And so specifically, we'd expect a plot that looks something like this, where on each of the axes here, I have the effect size for the genetic effect estimated in a given dietary environment. And again, if those effects were going in opposite directions, we'd see a negative correlation, such that alleles that are beneficial on the standard or the control diet are instead detrimental on the high sugar diet. However, when we actually look at our real data, we find that that's overwhelmingly not the case. And for most of the variants that are significant on both diets, the risk allele is always the same allele on both diets. So now what about that second pattern that I said we might expect under a mismatch, where there are alleles that just don't affect lifespan on the standard control diet, but then decrease longevity on this high sugar diet? Well, we obviously find a lot of evidence that those alleles exist, given that there are 702 sites with effects specific to the high sugar diet compared to only 2 sites that show the opposite type of genotype by environment interaction. So this suggests that there's a lot of genetic variation for lifespan that's only revealed when flies are exposed to this diet that's outside of the range of what the species has experienced through evolutionary time. Ideally, we might also want to know whether the set of alleles that decrease longevity on this high sugar diet have also experienced some history of positive selection in wild flies. However, it's been really difficult for us to infer positive selection in this genomic data because it's relatively low coverage, but this is a problem that we're actively working on. As a proxy, sort of in the meantime, what we've been doing is to ask whether the set of alleles that specifically decrease longevity on this high sugar diet are more likely to be derived alleles that recently arose in this particular fly species, or that recently arose on the lineage leading to this particular fly species. And when we test for this pattern, we find that those 702 alleles that decrease longevity only on the high sugar diet are in fact more likely to be derived and recently evolved in fruit flies relative to null expectations that we had inferred from non-significant sites. So here you're just looking at the proportion of derived alleles that decrease longevity, and the black distribution is the null distribution, and the orange is the data for these 702 sites. We find the same sort of pattern when we look specifically at the variants that decrease lifespan on both diets. And we think that that fact just reflects the idea that any allele that's associated with lifespan has probably been under selection in wild flies, not only those that display genotype by environment interactions, though we do find stronger evidence for that class of alleles. And so putting this all together, we do think that this supports the idea of evolutionary mismatch. Namely, our data suggests that exposure to evolutionarily novel environments, and in this case, the high sugar environment, seems to sort of reveal fitness decreasing alleles that we wouldn't have otherwise ever seen in the population. And we also find that recently evolved alleles are more likely to be detrimental in this novel high sugar environment. And so I realize that flies are not humans, but I do think that these data are really exciting because they represent one of the few genomic data sets that are out there that's really trying to test the ideas and learn something about the principles of the evolutionary mismatch hypothesis at the molecular level. And going forward, I think that accumulating more of these sorts of tests in humans and really working out the molecular details of mismatch is going to be incredibly important, because there's a really long list of chronic non-communicable diseases that have been labeled as putative mismatch diseases. And for essentially every gene or every disease on this list, not just for cardiometabolic diseases, these diseases seem to have some environmental component, they seem to have some genetic component, and there seems to be a lot of variation across people in terms of their susceptibility. And this really points towards a role for genotype by environment interactions, and we just need to start to work out the molecular details. All right, so to summarize what I've shown to you in this talk, we do find evidence that over evolutionary time, the physiology of the Turkana has putatively adapted to the ecological and the dietary challenges that they face, in particular to water stress and to a diet high in red meat. We also find that when individuals of this putatively locally adapted genetic background move to novel urban environments, we find that their cardiometabolic health suffers. And then using data from a model system, we're able to show that there are many loci in the genome that are neutral and not associated with fitness, and in our case with lifespan, under species typical conditions, but where those variants then do become detrimental and lifespan reducing when the population is exposed to an evolutionarily novel obesogenic diet. And with that, I want to specifically thank the Turkana Health and Genomics Project field team, namely Charles, Francis, Simon, and Eric, as well as all the communities and the participants that we work with. I also really want to thank a lot of different people that have been involved in this research, especially in my postdoc advisor, Juliana Rolls, as well as the other directors of the Turkana project, Dino Martins and Joseph Kamau. And I want to thank my funding sources. And then finally, I'm just going to end by putting in a plug for my lab. So as Ian said, I'm starting my own lab this fall at Vanderbilt. So if you know of anyone that's interested in this sort of work, I'm actively recruiting postdocs and lab technicians. And with that, I can take any questions. All right.
Fantastic.
I'm going to stop sharing my screen just so I can see all of you.
That was really fantastic, Amanda. Tons of really fascinating work. That was the first time I had seen the fly stuff. Super cool. I have some questions. Does anybody else have some questions? If you do, just turn on your screen and go ahead and ask them.
Hi. This is so I'm not a bioanthropologist, but I was wondering. So I've heard that BMI is not considered a good marker of health more recently, and I was wondering why it continues to be used in studies like this. Just in general. I know that wasn't the focus of your talk, but I was just about that.
Yeah, no, that's it's a good question. I think that it continues to be used just because there is so much sort of like reference data out there, and because it's something that, you know, when you look at a list of biomarkers that for which we might want to do some comparison in the literature, or for which there's sort of like known cutoffs that we could use for saying this is bad, this is bad, this is good. So that's probably why we tried to, you know, collect as many. I think also it's easy to collect, especially in field settings. That's probably another reason. But yeah, so we've been trying to sort of balance the what's sort of easy and feasible with you know, other measures, and just collect as many as we possibly can. But yeah, I think that yeah, you're probably right that it's not the only one that we should be focusing on, for sure. Yeah.
I have a question about the Turkana data set. So you focused on measures of diet, and I'm wondering if you have data on activity, or if you think, I mean, maybe those co-vary, or do you think that activity is less important than diet in predicting these outcomes?
Yeah, I think that activity is probably wildly important, and we just don't have good data on it right now. So that's something that's like on the top of our to-do list, and that has sort of been disrupted by the fact that we haven't been able to do any field work, you know, for quite a while. But I absolutely think that diet is probably equally, if you know, at least equally important. And we started with diet just because it's something that we can easily, you know, we can do 24-hour recalls during our interviews, and to collect activity data. What we've been planning to do and sort of started doing, but then got interrupted, was to you know, have people wear accelerometers or wear some sort of fitness trackers for at least a week, so that we could get sort of a good measure of activity levels. And we just haven't built up a large enough data set to do much with it yet. But yeah, absolutely.
Look forward to seeing those data when they come out.
Yeah, thanks. Me too.
Any other questions? So one question that I have, Amanda, is that I was interested, well, it's sort of a two-part question. I was interested in the genetic separation between groups on the west and east side of Lake Turkana. And I guess the way that I interpret that is that the lake itself has been sort of a barrier to gene flow between those groups. Does that make, I mean, it's an enormous lake, but I never really thought about it as being some like that before, but is that something that makes sense to you? And I guess the second question that I had relates to this gene STC1 and alleles related to it or close to it, and that is so you have data, you have genetic samples from non-Turkana pastoralists that have pretty similar lifeways to the Turkana. Maybe you don't have enough samples from those groups to answer this question, but I'm wondering if you also see this, you know, higher frequencies of alleles in this gene in these other groups that are in a similar environment, despite the fact that they're like, I'm thinking about like these groups are genetically distinguishable, but it would be interesting if they had, you know, they share the same lifestyle, and maybe on this like, you know, functionally relevant gene they are similar. Is that something you can answer?
Yeah, absolutely. I'm going to start with, I guess I'll start with that question. So we definitely, in the other groups, in the other pastoralist groups that are sort of relatively closely related to the Turkana, we do see the same alleles near that gene. And I think that it seems to be sort of a set of alleles that are prevalent in general in this like pastoralist complex and in this group of this group that lives in sort of that northwest Kenya, Uganda region. Interestingly, I think that there hasn't been much work done on this gene and on that region because that region is actually like entirely monomorphic in Europeans, which is where most of the, genetic work on any sort of health related stuff gets done. So I think that's pretty cool. And then to answer your question about about gene flow, yeah, I think that the lake itself probably is to some degree a barrier. It's also just been shown over and over again in other populations, not just here, that that genes often mirror geography. So sort of famously, if you genotype everybody in Europe, you can actually like recreate a map of Europe by like plotting everybody on like plotting people from all the different countries on a PCA, and you'll like recreate a map of Europe. So there seems to be a lot of fine scale structure in humans. I think we sort of don't know to what degree it's based on like, oh, there's a lake there, I can't go over there, versus just, home ranges or other sort of cultural and ecological factors that keep people sort of relatively tied to a given location. But it'll be interesting. We're trying to do, like I said, some more sort of formal demographic inference on these genomes to sort of understand how all these populations are related through evolutionary time and when there were splits between particular populations. And so I think that'll be really interesting to look at too.
Yeah, I suppose I shouldn't read into it too much, but I couldn't help but think about like whether such a barrier could have existed in prehistoric times and stuff like that between populations, even really early hominins or something like that, thinking about sort of the effect that lake itself had on populations on the west and east side.
Yeah. No, that's interesting to think about.
Yeah. Does anybody else have any other questions? All right. Well, thank you very much, Amanda, for a really amazing talk, and thanks everybody for joining us. And everybody have a good break. Congratulations on being done with the semester. It was a difficult one, I know, for many people, and we made it through. So good job, everybody. And once again, Amanda, thank you very much.
Yeah, thank you all for listening. All right, happy holidays, everybody.